conocimiento se fortalece cuando los nodos logran interconectarse. Bienvenidos al primer simposio iberoamericano de mirmecología, versión en línea.
Okay, good morning everyone, and thank you for coming to this new AMA section. Today we have the pleasure to present Professor Christina Kwapich, who is an associate professor in the University of Massachusetts at Lowell. Professor Christina, thank you for participating, and you can start.
Thank you so much for inviting me. You've done a wonderful job organizing this conference, and I'm having a great time learning about all the student research. No hablo mucho español, entonces me disculpo, pero mi charla es en el inglés. Si tiene preguntas, use el chat, please. So, let me share my screen. So, can you see the PowerPoint?
Yes.
Great, wonderful. Okay, so thanks everyone for your patience while we got started. So my name is Christina Kwapich, and I'm an assistant professor at the University of Massachusetts, Lowell. This is my first year as a professor, so I'm just getting oriented in my new position. But I thought I would introduce myself to you and tell you a little about my career trajectory before I open the floor for questions in case any students are hoping to follow a similar path. So my research is broadly focused around how sociality shapes ecosystems.
Professor Christina, so sorry for disturb you. You
No
can
problem.
put the presentation in a presentation mode. We are using the screen in PowerPoint.
And we'll do it this way. Are you seeing my presentation now? Got it?
Great, yes.
Okay, great. So as I was saying, we are ecosystem engineers in the same way that ants are ecosystem engineers. Both of us affect our entire landscape and all the species living there. So many of my questions about ants actually focus on how the ants affect the ecosystem they live in, from the soil to the other animals living in their nest to the plants that surround them. But I'm also interested in how the environment and ecology actually shapes the ant societies and the organization of those ant societies themselves. I'm a behavioral ecologist, so most of my work takes place out in the field. I believe that nature provides a context for behavior and life history traits, and in this way, we can better understand how these traits evolved. So a lot of my work takes place in the southeastern and southwestern United States in these beautiful habitats. And during my PhD work, I investigated the division of labor in this beautiful ant. This is Paganomermex badius, the Florida seed harvesting ant. And I enjoyed working with it because it not only makes very deep and beautiful nests, but it has this sort of interesting seasonal cycle of labor allocation that it follows. On the graph below, you can see how the allocation to foraging for seeds changes throughout each of these annual cycles in a very similar pattern. I worked with Walter Chinkel during my graduate career, and much of his work focused on nest architecture, which also became an interest of mine. Here we are out in the field casting an ant nest with molten aluminum, and this is the type of ant that makes the nest there on the left. I think field biology is particularly important because it can reveal things not only about ants, but about ourselves and other creatures living in the environment. For instance, in one study I did during my graduate career, we looked at how ants move soil up and down in the soil column without bringing it to the surface. So we discovered that Paganomermex badius actually uses a bucket brigade and caches great amounts of sand in chambers below the surface, sometimes not ever returning it up to the surface. So when they excavate their nests, they stuff a lot of sand in unused chambers. Because of this, sand from different historic stratas is mixed all up and down the vertical breadth of the nest. And this actually affects the aging of human cultural artifacts, such as this pottery shard here shown on the right. And so we were actually able to show that over millennia and even over a period as short as 100 years, the ants significantly disturb the soil column, which can affect our understanding and quartz clock dating of cultural artifacts. So as I've mentioned, a lot of my interest is in what goes on inside subterranean ant nests. And most recently, I started working on an unnamed species of Myrmica, which is found in the southwestern United States. And this species actually harvests pine resin from pine trees found in these ponderosa pine woodlands. My collaborator, Jeff Sosa, and I found that the ants hoarded the resin in their nests and actually process it down into little chips, which they use to tile their chamber floors. Here's a picture of one of the Myrmica chambers, which has been excavated. And you can see the brood there laying on top of these chips of pine resin. Of course, pine resin is full of terpenes, and we're actually interested in studying whether the pine resin in this case is used as an antimicrobial or to fight intimate pathogen and fungi, just as it's used in Formica paralugubris in Europe. Ants also create living nest architectures. Those of you familiar with ants that use black yeast in their carton nests may be interested to learn about some undescribed uses of lichen in ant nests in the southwestern United States. Here I've pictured several ant species, including Chromatogaster imbriana, and another Myrmica species, which have incorporated living lichen into the walls of their nests. I'm also interested in Lymotopene Mepiculatum and these wonderful trabecula structures they've built, which seem to be innervated by a living fungal partner. Here again is another picture of Chromatogaster building its nest wall, and the use of lichen in ant nests is actually a topic of my new PhD student's dissertation. Another of my interests, of course, is in the nature of seed storage and seed processing by seed harvesting ants. And my favorite Preguntame expedius is actually dimorphic. So you have large workers and small workers inside the nest. And for decades, we all believed that the large workers served the function of cracking open seeds. But in fact, in a study I conducted with Walter Chinko, we found that the large workers were no better at opening seeds than the small ones. And in fact, most of the seeds the ants collect stay in the nest for a year or more. And the ants need to wait for those seeds to germinate in order to access the seed pulp inside. The ants move the seeds up and down the vertical strata of their soaking wet nests and actually just try and, we believe, promote germination in order to access that seed pulp in times of starvation. However, storing lots of seeds in a soaking wet nest causes other problems, and the seeds can mold or spoil. So with my collaborator, Katrine Kellner, we're looking at the surface compounds on seeds and considering whether ants may apply any antimicrobials to protect their seed stores. And in fact, in a pilot study, we found that the seeds of Preguntame expedius are covered in actinobacteria, which have been implicated in antimicrobial uses for other kinds of ants. Another seed harvesting ant that my lab focuses on is Veromesser purgandii. It lives in the Sonoran Desert in Arizona. These ants are also polymorphic. They have a range of worker sizes in their nests. And this beautiful photograph is by Alex Wild. And I continued some of Steve Rissing's work, which looked at the effects of this polymorphism on other colony level traits. And what I found was that by measuring head width as a proxy for body size, we could see a trade-off in worker number and worker size for a select set of colonies in any given habitat. And those colonies, like the one on the right, which chose or by self-organized processes made more workers that were smaller, actually had fewer patrolines in their colonies. So the queens either mated with fewer males, or these colonies were older and the queens had used up sperm from their initial polyandrous mating event. In all, I was able to estimate how much biomass this ant species puts back into the desert each year. And in fact, for each hectare of Sonoran Desert, about 3.5 kilograms of dead worker biomass are returned annually. And that's equivalent to these numbers of these common desert animals, but also to approximately 1 human infant per hectare. So these ants are actually pretty incredible, and they have large effects on the environment where they live. Because these ants are polymorphic, I was curious whether the polymorphism that they exhibit might affect things like their nest architecture. And so to do this, I designed an experiment in my living room in Arizona and studied how nest building proceeded in groups of like-sized or groups of mixed workers to determine what nest structures they actually built. So here's just a short video clip of a nest cast made in wax at the end of one replicate of this experiment. And what my collaborators and I found was that when you place large and small workers together, they actually build more complex nests and deeper nests than workers of individual size classes of the same numbers placed in isolation. So there is an emergent property or an interactive effect of working with ants of different sizes. And we believe that this is because large ants are working on the digging faces of tunnels and pushing their smaller sisters aside, which ultimately leads to the production of additional shafts and tunnels. But body size has another consequence for the ants, and it's interesting to think about these colony-level tug-of-wars over body size and how they affect colony functioning at many different levels. In fact, Veramesta purgandii is one ant that participates in one of 4 types of rescue behavior that have been observed for ants. These ants actually rescue nestmates from the spider webs of Stetoda and Asaginna spiders, which are specialist myrmecophagous spiders. And it is the large workers only who respond to the alarm pheromones produced by those workers that are captured in the spider's webs. So again, there's a second function, or rather a third function, for large workers in the nest, which has nothing to do with architecture or territoriality and division of labor. Now, this behavior is very interesting, but because I'm a behavioral ecologist, in my lab, we like to quantify the costs and benefits of behaviors like these. And so we determined that ants bring in an average of 2 seeds per day, and that their foraging tenure, or their career as a forager, is about 18 days. Given these data, we were able to estimate that if spiders consumed 5 ants from a colony per day, that the colony would ultimately lose 65,000 seeds per year. So there's a high cost to spider predation, which could explain why a complex and sort of puzzling behavior like this might evolve. Typically, we see rescue behavior in species with very small colonies where individual value is high. But we can also see how it would evolve in a large society like this one. The final thing I'll mention is that I'm very interested in myrmecophiles and myrmecomorphs, so the guests of ant colonies and creatures that mimic ants. So ant nests are really like ecological islands, and all of the creatures living inside are subject to the effects of the ecology of the ant nest. I'm particularly interested in generalist myrmecophiles. These are creatures that can live with more than one species of ant host. And in particular, I've been working on myrmecomorphic spiders and myrmecophilous ant crickets. So in the southwestern United States, Myrmecophilus manni, this ant cricket, actually lives with up to 41 different ant host species. And depending on which host species it lives with, it takes on a completely different phenotype, and in fact varies about 2-fold in body size, or actually 2.5-fold. So very small crickets live with small ants, whereas large crickets live with large ants. Why this occurs, we're not sure, but we've done cross-fostering experiments and can actually induce this change in body size just by cross-rearing them in different ant nests. Together with my collaborator, Jeffrey Sosa Calvo, we're working on constructing a cytochrome oxidase 1 gene tree to determine if there are actually any cryptic species in this system, because there are certainly trade-offs associated with body size within Myrmecophilus manni. For instance, both large and small crickets lay eggs of exactly the same size. All of their eggs are 1.2 millimeters long. So the investment that a small cricket has to make in a single egg is actually much higher than a large cricket. And what we actually see here is that Myrmecophilus manni is probably best described as a species complex, but within individual sites, all the crickets exploit multiple hosts, and they come in all of the body sizes that we see. So there's both cryptic species and phenotypic plasticity in Myrmecophilus manni complex. So I've been chatting for a while now about my research interests, but the topic of this talk is actually Ask Me Anything about being a new professor. And so I'll just speak briefly about my experience in the last year. Of course, I was inspired, like many of us, to study ants by these two great books by Bert Holdobler and Ed Wilson. And I had sort of a strong interest in lizards and in ants, and these books really tipped me towards the ant direction. And what I've learned over the years is that in order to study ants on an academic path, you have to be rather mobile. You need to be nomadic, just like an army ant. So I was born in the Midwest of the United States in sort of farm country, and did my undergraduate not too far away at The Ohio State University, where I majored in entomology. There, I worked with Joan Herbers on acorn ants, Timnothorax curvispinosus, and I also worked with Susan Jones on termites, Reticulotermes flavipes. I met a great mentor there named John Wenzel, who recommended I go to graduate school with Walter Chinkle at Florida State University, and it was an excellent match both in habitat and in personality with my advisor, and we had a great time. I really enjoyed my graduate career. After 6 years, I started a postdoc with Bert Holdobler at Arizona State University in the Social Insect Research Group, where I continued my work on myrmecophiles and seed harvesting ants. Now, after 5 years, I've just started my assistant professor position at the University of Massachusetts in the snowy northeastern part of the country. So what I just want to point out with this is that academia would seem to break your allegiance with every place, every group of friends, even your family, because you have to move so often and you often don't get to choose where you move. But I would say that rather than breaking the connections between all of these places, I've just accumulated more connections. And when you move for academic jobs, you can always go back and find colleagues and friends wherever you go. So it's sort of a nice thing, I think. If you're a student, the next thing I would recommend is to choose your advisors wisely and prepare well for any opportunities that might come your way. I was extremely lucky to have 3 very kind advisors at each stage of my career, and they invested in me heavily, and they continue to invest in me now with collaborations and mentorship. I had many other mentors along the way, which are not all pictured here, but I think the best thing to remember is that you have value, and you know, you can always move if you're not happy where you are. I lucked out, and luck is also a big part of the game. So I'll just say that I met Bert Holdobler, my childhood idol, at a conference, and it happened to be the last day of the conference, and I bumped into him at a poster session. So it wasn't unlike this virtual conference, and because of that single interaction, he invited me to be a postdoc 4 years later. So there are serendipitous chances that can happen and can change your life for the positive, and I have to say that by and large, my experiences have been positive. Now I'm at the University of Massachusetts Lowell, but it wasn't easy to get a job, and those of you in the postdoc stage or other stages know that that's the case, and it's increasingly difficult to find a job in academia. Over 2 years, I applied to 34 different jobs. I had 4 phone interviews, 2 campus interviews, and only 1 job offer, and it was here. But I'm very glad it was at UMass Lowell because it's a research university, and I actually really like my colleagues and where it's situated. So when I joined the department, there were 12 male faculty researchers and 4 women. And although this disparity seems like it might be troubling, actually the most difficult thing I've had to overcome was the fact that there are no other behavioral ecologists in my department. In fact, most of my colleagues study biochemistry or cell and molecular biology for the purpose of human health. And so coming from a research group that was focused exclusively on social insects, this was a big change for me. But in fact, these are all wonderful and curious people, and they're simply bowled over when I tell them things like all the ants in the colony are female. So it's very easy to impress them with ant facts and they're eager to learn them, just as I'm eager to learn about what they do. The department has lots of undergrad, master's, and PhD students, and I just invited my first PhD student this year. His name is Hoon Kang. So on this note, I would just say, be true to your identity and find a gap you can fill. I was hired in this department because they didn't have anyone teaching behavioral ecology or entomology, and they wanted to expand and broaden the goals of the department. So I was lucky to find this niche. Here's a picture of my lab a couple months ago before I started filling it up. We have a small and basic molecular bench, and then also a lot of space for behavioral experiments, and a USDA certified ant room, so we can invite exotic ants from all over the world to stay in the laboratory. It's an environmental chamber with temperature and humidity control, so hopefully I can go in there during the cold Massachusetts winters. I also have some research students in the lab, and it's been fun to get started despite all of the COVID-19 issues.
Professor Christina, right now we have just 5 minutes for questions.
Okay, this is my last slide, so I will just finish here, and thank you for your attention. So please ask me any questions you might have, and I'm going to stop sharing screen so I can see you guys, okay?
Great. We have a first question, and it is, how can we balance a nice amount of these awesome facts and experiments we want to share as scientists and the mandatory class program?
That's a good question. I would just say get your courses done early if you're in graduate school, and try and save your field seasons for field work. But it is difficult to balance these things, especially when a PhD program may be shorter. So I always have one big focal project and then small ones that go with it. And sometimes those small ones develop later on. And many of the things I'm talking about today, I had ideas about in grad school, but didn't have time to do. So you have to bide your time carefully, but yeah.
Great. We have the other question. What are the best qualities a mentor can have, in your opinion?
That's a good question. I would say patience, ability to let you conduct your own research independently without managing everything you do. The purpose of graduate school is to become an independent thinker and creative researcher. So you shouldn't be working for your advisor on their project or their interests all the time. You can certainly help them and learn from them, but you're an independent scholar and they should allow you to maintain that independence. They should also invite you to work with them named on papers if you participated, and they should allow you to be first author on any of the papers where you did the bulk of the research. So having research ownership and independence, I say, would be a really important factor. And it's hard to strike that balance between guidance and independence, but the best mentors do it well.
Great. I don't see any more questions in the question and answer section, but I have a personal question. What is your advice for those postdocs that are currently looking for job positions?
Yeah, well, I would definitely recommend using the Evo Job Wiki. It's a wiki online, which you're probably familiar with, which posts a lot of jobs around the world, but mostly focuses on evolution and ecology in North America. And also in your cover letter for your job, in the first paragraph, I think you should present a list of your products. You want to say, since 2015, I've published 5 papers, I've applied for 2 grants, and I've traveled to 2 continents to conduct field work. And really just, list everything and market yourself like you're selling a product, because those people reading all the applications, sometimes they don't even get past the first paragraph before they move on. So really try and cram it in there and identify yourself. Say, I am a behavioral ecologist interested in social organization, or I'm a molecular biologist doing this. So you have to think of your moniker and your tagline and how you define yourself. And that actually might change for each job you apply to. So you need to tailor it very carefully to each job.
Great. We have just time for one last question. This is from Corina Barrera, and she said, "Hi, Christina. Thank you for your presentation. Very nice. My question is, how is it to work with the great dressers, Berthold Doppler?"
Wow, it's an honor to work with him, and it's incredible what a fast and competent writer he is. He writes on yellow notebook paper, and he includes citations and everything, usually in the first or second draft. He can write pages and pages and pages every night. So it's really incredible to see that process and how incredible his mind is. But overall, he's very kind and generous, and allowed me to pursue whatever questions interested me when I was working in his lab. So it was a great friendship. Yeah.
Professor Christina, thank you very much for this presentation. So right now I'm going to start with the roundtable. So thank you very much.
Appreciate it.
Para más información, visita nuestra página web o comunícate a nuestro correo electrónico.